Our next speaker this morning is Dr. Alexa Adams, who's Assistant Professor of Pediatrics at the Division of Pediatric Rheumatology, Weill Cornell Medical College, and Assistant Attending Pediatrician at the Hospital for Special Surgery in New York. She received her bachelor's degree, University of Virginia, her medical degree, Medical University of South Carolina. She was an intern and resident at New York Presbyterian Hospital, a fellow at Pediatric Rheumatology at Hospital for Special Surgery. She is now the Associate Director of the Pediatric Rheumatology Fellowship Training. She was awarded the Distinguished House Staff Award from Cornell Medical Center. She has numerous publications, and this morning she's going to talk about celiac disease and rheumatology.
Okay. So good morning. My name is Alexa Adams, and as Dr. McLean said, I'm going to be discussing celiac disease and rheumatology. But first, I'd like to thank the Congress of Clinical Rheumatology for their kind invitation to come down to Florida for a little while and have this opportunity to talk to you about a topic I find extremely interesting. I am, what? Oh. Can you hear? Yeah. The green light is on. Is it? Okay. All right. I am on the Speakers Bureau for Abbott Pharmaceuticals, but I don't have any relevant disclosures to today's topic. So in considering this topic, I decided to approach this talk looking at celiac disease both as an autoimmune disease and in association with other autoimmune diseases. I plan to touch on the genetics and pathogenesis of celiac, including the contribution of the impaired mucosal barrier. We'll review celiac antibody testing, the wide spectrum of clinical celiac disease, the need to screen for celiac, and the risks of untreated celiac. And then finally, we'll end with some illustrative clinical cases. So celiac disease is an immune-mediated enteropathy triggered by the ingestion of gluten in genetically susceptible individuals. And the presence of HLA-DQ2 and or DQ8 are necessary but not sufficient for the diagnosis of celiac. Celiac is a permanent autoimmune condition, which is unique among the autoimmune diseases in that both the trigger, that being gluten, and the autoantigen, that being tissue transglutaminase, are known. And treatment, of course, requires the permanent dietary elimination of gluten. Celiac affects 0.5 to 1% of the United States population, and a large U.S. epidemiologic study demonstrated the presence of celiac in about 1 in 130 healthy subjects, in about 1 in 20 first-degree relatives of known celiac patients, and in 1 in 40 second-degree relatives. And there's a well-known increased risk in Downs, Turner's, and Williams syndromes. The autoimmune features of celiac include the production of disease-specific IgA and IgG antibodies, autoantibodies to tissue transglutaminase, direct cytotoxicity of intestinal intraepithelial lymphocytes on enterocytes expressing MHC molecules, a genetic predisposition and an apparent multifactorial etiology, and a coexistence with other autoimmune diseases. Celiac is unique among the autoimmune diseases in that early identification and treatment may actually reverse damage, and it may prevent long-term complications and comorbidities. And it's also unique in that the treatment does not require any medication, so treatment of celiac carries almost no risk of adverse side effects unless the patients are following some sort of very extreme diet, which isn't recommended, above and beyond the gluten-free diet. Just to touch on the genetics briefly, again, HLA-DQ2 and or DQ8 are necessary for the development of celiac, and about 30% of the U.S. population is DQ2 positive. And of these patients, fewer than 2% will actually go on to develop celiac. There's a high negative predictive value in that the absence of DQ2 or 8 excludes celiac with a 99% confidence. There are other non-HLA genes which are contributing. They influence different aspects of the innate and adaptive immune responses, They influence intestinal permeability and general predisposition to autoimmunity. And while several of these have been identified, their influence has not yet been confirmed in celiac. So celiac is being recognized with increasing frequency throughout the world, and with the advent of serologic testing, there's been increased celiac case finding. There are two areas of particular interest, one being India, where the HLA-DQ2 and 8 genotypes are common, and where wheat consumption is high. And with the serologic testing, there's been more case finding here. And also in other parts of Asia, particularly China, where there's been increasing use of wheat-based products. So in the coming years, we can expect to see more data on the prevalence and clinical implications of celiac in those populations. Celiac is activated by proteins in wheat, barley, and rye. And while gluten technically only encompasses the proteins found in wheat, those being the gliadins and the glutenins, there are other closely related proteins, the hordanes and secalins, which are found in barley and rye, and those can also activate celiac. And if you just look at this plant taxonomy diagram, you can see that wheat, barley, and rye all have a common ancestry in the grass family, which is why there's overlap in these protein structures. So all of these related proteins have a high proline and glutamine content and are resistant to degradation by the various digestive enzymes. Large peptides then accumulate in the small intestine, and tissue transglutaminase, which is released by the intestinal mucosa during times of injury or inflammation, has a high avidity for these peptides. It deamidates glutamine, which is present in high content in these peptides, and converts it to negatively charged glutamic acid. The deamidated gluten then has an increased affinity for the DQ2 and 8 molecules, and then this complex can activate T cells in the mucosa of the small intestine. With that, interferon and other cytokines perpetuate an ongoing inflammatory response. IL-2 and 15 have been strongly implicated in this. Damaging enzymes are released, including MMPs, and all of this culminates in intraepithelial lymphocytosis, loss of villus structure, and crypt hypertrophy. And this is a cartoon depicting this process, but I think it's actually a useful one because it enlarges this complex and shows you how in the presence of tissue transglutaminase, you have conversion from the neutral glutamines to the negatively charged glutamic acid, and this complex together helps to activate the T cells in the intestine. So in addition to all of this going on, it appears that mucosal injury and loss of barrier function is key to the development of celiac autoimmunity. The risk of celiac depends on the host's encounter with gluten peptides at a time of intestinal inflammation. And this provokes interferon production, antigen-presenting cell activation, and a Th1 cell response. And it appears that mucosal infections may in fact set the stage for this, with accompanying tissue damage and release of TTG. So there's some interplay between adaptive and innate immunity here that's probably responsible for this celiac autoimmunity. This concept of the leaky gut has been proposed as key to the pathogenesis of a number of other GI and autoimmune diseases, including some of those that are depicted here. And unfortunately, this is not projecting very well at all. But in addition to all of these, a number of other rheumatic diseases, including the spondyloarthropathy spectrum diseases, have been implicated in the leaky gut model. At the same time as all of this is happening, people have been looking into the early events in celiac disease, and a protein called zonulin appears to be upregulated. This is an endogenous protein that's functionally and immunologically related to the zonula occludin toxin from Vibrio cholera. It's a modulator of intestinal permeability, and it induces tight junction disassembly. So it's been hypothesized that this is an early change which precedes the onset of gliadin-induced immune events. And this group, led by Fasano in Maryland, showed that with removal of gluten, you have a decrease in zonulin levels and a normalization of the intestinal barrier function. A number of environmental factors have been investigated in the pathogenesis of celiac, The jury is really very much still out on most of these, but just to mention them, the duration of gluten exposure may have a role, and I'll come back to that when we discuss the autoimmune diseases specifically. Some observations